Now next chapter on anatomy and physiology in sexual medicine. This chapter will discuss those aspects of anatomy and physiology that are relevant to the sexual activity, and will skip the basic information that is taught in medical college. Male anatomy. During the first 2 months of embryonic development, there is no much difference between the male and female sexual structure. The embryonic phallus becomes penis of the male or the clitoris of the female. The adult structures are richly supplied with the nerves which terminate in end organ of touch called Meissner's corpuscles. Corona, the lower edge of the glans, is the area that is the most sensitive to tactile stimulation. The frenulum under surface of glans is similarly sensitive to touch. Frenum is the longitudinal fold by which the loose foreskin is attached to the glans. If the fold is not removed by circumcision, it is advisable to preserve the frenulum during circumcision. This is a minimum of sensation is the skin covering the main shaft of penis. Penis consists of 3 parallel cylinders of spongy tissue bound in a thick membranous sheet. The upper 2 are corpora cavernosa, and the one of the underside of the penis, through which urethra passes, is known as corpus spongiosum. Penis has numerous blood vessels. During sexual arousal, the blood rushes in the cause erection of penis. The head of the penis, called glans, consists of corpus spongiosum and a highly concentration of sensory nerve endings than the shaft of penis. The skin covering the penis is freely movable at the tip and is called a prepuce. The cherry substance called sphegma is secreted by prepuce and is accumulated underneath the prepuce. Sphegma should be washed away while taking bath. The color, size and shape vary considerably from one male to another. Smaller non-erect penis usually becomes larger when erect penis and vice versa. Though the penis is considered as an organ of masculinity by many, in reality the function of penis is like a dropper to carry the sperm from the body of male to the mouth of uterus of female for fertility. Penis loses its erection when the job is done. The average size of the erect penis is about 13 cm 5 inch, and the girth circumference is about 1 to 1.5 cm, max 4.5. However, penis of the size of a little finger is taken as normal, since it equally serves the purpose like any other penis, so the size doesn't matter. Testis is composed of coiled seminiferous tubules where the sperms and sperms are formed. Sperms then empty into epididymis, a 20 feet long coiled tube. Epididymis lead to vas deferens, which enlarge into ampulla immediately before it enters into the prostate. Seminal vesicle located on each side of the prostate empties in the prostatic end of the ampulla, and the content of both the ampulla and the seminal vesicle pass into the ejaculatory duct leading through the body of prostate gland to empty into the internal urethra. Prostatic ducts in turn empty into the ejaculatory duct. Finally, the urethra in the penis is the last connection link from the testes to the exterior. A large number of small glands of litter located along the entire length of urethra and the bilateral bulbourethra. Copper's gland, called a Copper's gland, and produces mucus that is added to the semen. How the spermatogenesis occurs? In the seminiferous tubules at about the age of 13, as a result of stimulation by gonadotrophic hormones, spermatogenesis continues throughout the remainder of life. Following formation of spermatozoa, they enter into the epididymis. After 18 hours to 10 days, sperm develop the capability of motility and maturation that enable the sperm to fertilize the ovum. Most of the sperm are stored in the vas deferens and ampulla. Sperm can be stored there for a month, maintaining their fertility. Normal sperm travels 1 to 4 mm per minute in a straight line in alkaline fluid media. Sperm remains alive for 1 to 3 days in the female genital tract. Seminal vesicles are lined by secretory epithelium that secretes mucoid material containing fructose, prostaglandin, and fibrinogen. Secretion of seminal vesicle provide nutrition to the ejaculated sperms. Prostaglandin aids fertilization. Prostate gland secretes the thin milky alkaline fluid containing citric acid, calcium acid phosphatase, and clotting enzyme and fibrinolysin. During the emission, the capsule of prostate gland contracts simultaneously with vas deferens so that the thin milky secretion of prostate gland is added to the bulk of semen. Prostatic fluid neutralizes the acidity of the other fluids after ejaculation and greatly enhance the motility and fertility of the sperms. Semen is composed of the fluids from the vas deferens, seminal vesicles, prostate gland, and from bulbourethral glands. Prostatic fluid gives the semen milky appearance, while the fluid from seminal vesicles and bulbourethral glands gives the semen a mucoid consistency. The clotting enzymes from the prostatic fluid and fibrinogen from seminal vesicles form a coagulum which is dissolved in the next 20 minutes. by fibrinolizing from the prostatic fluid. Sperms remain immobile for some minutes after ejaculation. However, after about 20 minutes, sperm become highly motile. Therefore, women desiring fertility should remain supine on the bed for about 30 minutes after the coitus, especially during the fertile period. Erection of penis. Penile erection of penis we will discuss in the next